Yes, part 2. You came back for more. Dare I say, this one's a lot shorter. I say that and then it isn't, so I'm going to say it anyways. We're going to finish that last part of our nucleic acids. In the last lecture, we learned all about the structures, the phosphates, the sugars, the bases that come together, more of like the biochemistry, right, on the chemical level. Now we're going to look at the last 4 slides, which we're talking about RNA, how it's structured, the differences, and then those really important scientists, right? So we left off here, and now we're going to finish these last things, the RNA, Watson and Crick, and how DNA is organized in your cells, okay? All right, Miss Myers, look at all this stuff that you know. Let's start here. Okay, all right, so we learned all about the R's of the DNA, and so now we're going to look at those 3 different types of RNA, the ribonucleic acids. Remember that? These are single-stranded, right? So I just get 1 strand versus the 2. Remember that their function is temporary, like a temporary little message. It's going from the nucleus and out to the ribosome to make the proteins. And then we have 3 different types of RNA that you'll need to know about, and we'll learn more about that with our protein synthesis later, right? The RNA, oh my gosh, is usually single-stranded. Oh goodness, when it's forming the complementary bases, it loops, right? So it can loop around, but we're still not getting the double helix coming together, right? My 3 main types, I've got my mRNA, my tRNA, and my rRNA, right? So messenger, transfer, and ribosomal, all 3 of those. This is my messenger RNA. I love when the actual name of the thing matches with what it does. The messenger RNA is the little thing that goes into the nucleus, and it transcribes or writes down just part of a gene, right? It's looking at just that little gene, and it takes it out to the ribosome to make my proteins, which we learned about last time. Those little codons, right, go for my amino acids, right? So it will copy down ATC, right? AUC. And it takes that code and it leaves the nucleus, right? So it's allowed to go out of the nucleus into the ribosome to make those proteins. Okay, so that's my messenger RNA. My ribosomal RNA is the catalytic. What is catalyst? Where have we learned a catalyst? No? All right. The RNA is the catalytic component of the ribosome. So it's the one that starts the reaction. Remember, catalyst is the impetus that starts the reaction. So this is the one, it's the component of the ribosomes that's going to start that process, right, to start those chemical reactions. And then my last one is my transfer RNA. My transfer RNA takes this little amino acid, right? Here's my amino acid here, and it's going to transport it to the ribosome. So it transports amino acids to the ribosomes. So this little messenger RNA here pairs up with the transfer RNA, and it just drops off the amino acids and puts them together with those peptide bonds. Yeah. So these are my amino acids that eventually come together, and that these base pairs here are complementary to those. So those are my 3 different types of RNA. And you should need to know the function of all those, and then we'll look at those much more in depth when we look at protein synthesis. All right, let's talk about some famous old white guys. Okay, we'll talk about how they stole the work from women. But anyways, DNA, Watson and Crick. So Watson and Crick are given credit for figuring out that the DNA has a double helix, right? This is in 1953. So these are the people that figured out that DNA is actually, right, in this type of structure. Ooh, it's lunch. I need to go get some lab supplies. And they're the one, they did this with X-ray photography, and they are the ones with, or I want to say, like, given the credit for figuring out that it's the double helix. Okay. Their efforts were guided by these people. Franklin, right here, this is the woman right here they stole from. She shared this, right? Or she actually, I'd have to look at the video again, but it was basically like she worked in the same lab with these 2 guys, right? And she was doing the same thing, this X-ray photography, and she also found the helical structure, and they shared her information without permission. This actually brought up a lot of like ideas of like working in the same lab and like who has the rights to permissions to things. And she was actually not awarded the Nobel Prize because she had already passed away by the time these 2 gentlemen actually got that. Okay. Some other scientists that we'll need to know, we're going to learn about Chargaff and his base pairing rules. That is in your book. and looking at how he figured out the base compositions by looking at purines and pyrimidines and how they have the same number. Levine was the one that was attributed with the nucleotide, that they are made of these 3 different parts. And Pauling is looking at the bonds, right? So the molecular distances between them and all these. So all of these 6 people together kind of put up the Euclidization of DNA. So all those different parts that we learn about now, the model of DNA, are put together from a lot of different scientists with some scandal and some drama, right? In your book, we're going to look at Chargaff's base rule and do some calculations for base pairing and how we figure out there are those different bases in each. Okay, all those things put together, this is like a physical model. So without looking at all those, colors and pretty things, this is what physical looks like. I can see here, right? I can see my, what are those called? Sorry, the sugars on the outside, the backbone kind of goes like this. In the middle, I can see these nitrogen bases come together. But they're the ones, like, these all people came to put together that these are my basics for my DNA. I've got my anti-parallel DNA strand, so the 5 to 3, 3 to 5, right? I've got my outer sugar right here, my phosphate, they're put together with those phosphate bonds here. Here are my sugars. I've got my bases, the rungs here in the middle that are complementary, right? And this overall shape forms a double helix, and the importance of that double helix for fitting that much DNA in a small part. So that is our current, there we go, model of a DNA strand. Okay. Hold on. Back, mouse. There we go. Okay. Haha. Okay. You need to watch me go around here. All right. So how it's organized, right? How it's put together. This is our last slide here. So prokaryotes and eukaryotes, we've already, again, talked about this. Now we're making a little bit more sense. In my prokaryotic, the DNA is naked. It's not bound to any proteins, right? It kind of makes this circular formation. Remember that there is no nucleus around it to protect it, kind of free floating through the cytoplasm. In eukaryotes, it's linear but bound to histones, right? So here is my DNA here. And eukaryotes, it's bound to my histones, which is a protein. We're very learned about histones. So mitosis. And as it starts to form, this chromatin condenses into chromosomes during cell division. So remember that in mitosis, when we talked about, right? So like at the beginning in interphase, the DNA is kind of just all over the place. It's still wrapped, right, in my helix, but it's all over the place. But when it goes to divide, remember that it doubles its DNA, right? So we learned about like interphase, it doubles DNA, and then it goes into a process where chromosomes become visible. Right? So at that very beginning, I get those meeting in the middle, right? And then I get the chromatids pulled apart. So each one of these chromosomes, and we'll look at this in genetics, contains a single DNA molecule. And the chromosomes here are like split up this way and that way, right? So we've already learned a little bit about how DNA becomes organized before it doubles. But now you can actually see, like, this is my prokaryotic DNA, this is my eukaryotic, very long strand, right? In my eukaryotic, the DNA tend to be a lot shorter. Okay. All right. End of section review is this, and I'll make sure I type these up. Can you do the following, right? This was my part 1. This was my part 2. We'll do some Chargaffs in class. We'll do some how do you actually draw these things so that the people give you points for it. And the other thing we'll do in class, I wanted to show you. So here's our Chargaff's rule. There's our drawing. Looking at the bases and how we can actually look at those with our chemical eye. So we've seen those. All right. Okay, that's the end of topic 2.6. I will see you in class. Have a fabulous day.